Good morning. Thank you for again, and thank you for the introduction. Today, I will talk to you how in our research here at Wageningen University, we use atomic force microscopy and a combined approach with vibrational spectroscopy in order to unravel the morphological, structural, chemical properties of protein self-assembly. Why this is important? This is important because we want to understand life. We want to understand how cells works. In order to do so, we need to understand how the biomolecular machines at the interior of a cell are performing their function. As already Feynman stated at the beginning of the century, it can be very easy to solve fundamental biological questions. We can just look at the thing, where the thing is this single biomolecules, such as proteins, and their self-assembly state. However, this is not so easy because not always we can look at the morphology of an object to understand how it's delivering its function in cells, but we have to care of its chemical properties. And so we need to understand an intrinsic correlation between the chemical structural properties of biomolecules to understand how they deliver their function in living organisms. The problem often is that this happens at the nanoscale, and so it's very difficult to visualize these processes. And this is not only important for health, but at the same way, this is important for disease. And this is because in the last century, a new class of human diseases has been discovered, such as neurodegenerative disorders, which are related to the aggregation of proteins. So what we have a morphological change in the shape and the state of a protein that goes from a single protein level to the aggregation into intermediate oligomeric species, which are spherical, into some protofibrillar species, and finally leading to the formation of fibers, which were called historically amyloid. However, what we also know that this morphological process, as I said before, the structure is related to a chemical change and a structural change. And this is related to the fact that during this aggregation, proteins undergo a misfolding, so a change of conformation that can lead to the formation of these fibers, which have a unique structure, which we call cross-beta sheet structure. And this is a series of intermolecular hydrogen bonds that stabilize beta sheets, which run perpendicularly along the axis of these fibrils. And again, these fibers have a diameter between 1 and 10 nanometers typically. So we need to be able to study them at the nanoscale. So in our laboratory here in Wageningen, what we do, we develop scanning probe microscopy methods, also combined with spectroscopy, as well as using microfluidics and sensors in order to study biomolecular processes. And one of the fundamental biomolecular processes we are interested in is the phenomenon of protein self-assembly. That is also related to a phenomenon we will see it later that is called liquid-liquid phase separation, which is fundamental for cell function. But then, as well as studying these materials in life and disease, we are interested in understanding how protein self-assembly can be used to produce functional biomaterials. So today I will talk about how we use AFM and infrared nanospectroscopy to reach this objective. However, before to do that, since I have a bit more time than a quick conference talk, I would like to say, but why AFM? Which single molecule technique we want to use, and how do we place ourselves in the field? And the reason is because bulk techniques, they can be useful to have very detailed structural, chemical, and kinetic characterization of, for instance, protein self-assembly of biomolecular processes. But these processes are, in most cases, very heterogeneous, as I showed you before. We have monomers, oligomers, and different kinds of fibrils. So it's not sufficient to look at average properties. We need to look at them at the nanoscale. And so, historically, microscopic methods working beyond the diffraction limit of light have been developed. And the first one historically has been electron microscopy. However, the technique requires conductive samples, requires staining, and it works in vacuum. As well as then a major advance has happened with the advent of cryo electron microscopy, which enables to evaluate the 3D structure of biomolecules with very excellent a resolution in the order of angstroms. However, because there is no staining, as in conventional electron microscopy, there is very low contrast. It's very difficult to see very small molecules. And this is not a true 3D technique. Another possibility is to use single molecule fluorescence techniques, and that enabled by using dyes to stain particular biomolecular processes and observing their dynamics. However, the resolution is limited to 20 to 100 nanometers typically. So finally, another possibility is to use atomic force microscopy. The technique, as I think you might know from this conference, works similarly as a person will do reading in Braille. There is a sharp probe, which is scanning the sample, similarly as a blind people will use his finger, to feel the interaction between the tip and the probe to retrieve its morphology. The high advantage of atomic force microscopy is that it can work both in air, in liquid, and in vacuum environments, so it's very versatile. And so normally, every one of these techniques has its own advantages or disadvantages, but another further advantage of atomic force microscopy is that it's not purely an imaging technique. Indeed, aside the enriching sub-nanometric morphology, characterization, atomic force microscopy can exploit this probe and the cantilever to acquire, between many other properties, nanomechanical properties. So for instance, to measure the Young modulus of a molecule, as well as can be combined, as we will see today, with infrared light or spectroscopic techniques to acquire chemical information at the nanoscale. So this is what is interesting for us to understand the characterized biomolecular processes in very high detail, with down to single molecule structural and chemical characterization. So I will talk today about multimodal single molecule characterization beyond imaging. What this means, let's suppose to do the following experiment. We have some protein fibres, as the one that I showed you before, involved in the onset of Alzheimer's. And, we have some nucleic acids, some DNA, and we take two separate test tubes, we deposit them on our surface, and we can image them with AFM, and we can achieve maps with very high spatial resolution. Then we can do another experiment. We take these two samples and we mix them together in the same test tube, and then we deposit them again on our surface. What we will notice that now it's much more difficult to distinguish what is DNA and what is protein in this map. And this is because we are chemically blind. A possibility to characterize our sample is to use, for instance, infrared spectroscopy, which is a technique which can understand the chemical composition of a sample by looking at the frequency of vibration of its molecular bonds. And we can see that we have indeed in the spectrum of our sample, both the amide band 1 of proteins, as well as the phosphate group of nucleic acids. However, the technique has no really spatial resolution at the nanoscale. And so basically we cannot see what is what in our map. The solution can be obtained if we combine nanoscale imaging with vibrational spectroscopy. And this is what we call infrared nanospectroscopy. This is a technique, as we will see in the following slides, that then enables to combine, to have simultaneously a morphological map and a chemical absorption map. Then what we can do, by pointing our tip on specific molecules on our surface, we can acquire infrared spectra of each single biomolecule. And by looking at the vibrations of the spectra, we can see, for instance, that this is DNA, while this is a protein aggregate. In the case of the aggregation of proteins, as I showed you before in the onset of neurodegeneration, then what we can do, we can use this amide band 1, which is related to the C-double-O stretching of the protein backbone, to study the secondary and quaternary structure of the proteins. And so to study how the conformation is changing during their aggregation in the brain of related to the process occurring in the brain of patients. And for instance, we can say that oligomeric intermediate aggregates, they have alpha helical conformation, while fibrillar aggregates, they have a beta sheet 1. And technique works following according to the following principle. if we are collecting an IR map, a morphology map, similarly as we do in any conventional IFM, and meanwhile we send IR light, if this IR light is absorbed by our sample, the chemical bonds will start to vibrate. And this vibration causes a thermal heating of the sample and a thermal expansion. This expansion can be measured by the cantilever in contact with the sample, and this response is proportional to the IR absorption. So this means that if we measure this response at every pixel of our map, we obtain a chemical map. Then, since IFM is also very sensitive for sensor, we can simultaneously have a nanomechanical map. If then we point at a specific location of our map and we sweep our laser as a function of wave number and we record the cantilever response, we will obtain a localized IR spectrum. And as I said, and we will see in the next slides, in the case of proteins, amide band 1 between 1700 and 1600 centimeters minus 1 is related to the C-double-O stretching of the protein backbone and is related to their secondary structure. So this means that now we can characterize simultaneously the morphology, the nanomechanics, and the chemistry of proteins. Now I would like to show you just very quickly a few breakthroughs that we have realized in AFM and in imaging and in characterization beyond imaging. We have started now several years ago where we have demonstrated the first application of nano-IR to and infrared nanospectroscopy to characterize protein droplets. And then we have combined the technique with microfluidics to reach single molecule detection, as well as to have liquid operation. And finally, very recently, we have demonstrated that the technique enables to characterize morphological and structural properties of single proteins at the nanoscale. And very quickly, how we achieved this. First, our interest was in achieving the capability of measuring in situ, so in liquid, because this is very important to study biomolecular processes. To do so, we used a setup of the system where we worked in a total internal reflection setup. This laser is sent from the bottom on our sample, and an evanescent wave is going into the sample at the surface with our proteins. And since there is an evanescent wave, the IR absorption of water is curved. And what we could show is that we could characterize the protein aggregates involved in the onset of Alzheimer's disease with a similar resolution in liquid as in air. And we could collect successfully infrared nanospectroscopy chemical maps of IR absorption. Then we could acquire localized spectra, and we could see that the spectra in air and in liquid were very well corresponding, aside for the fact that now we have an hydrated molecule compared to a non-hydrated one. Then what we could demonstrate is that if we will take fibrils formed by amino acids that have different chemical properties, we could obtain fibrils that are morphologically similar, but with a different secondary structure that we could recognize at the nanoscale. Then, of course, we wanted to say, okay, we can work in liquid still with some limited resolution, but can we go down towards the single protein molecule scale? And we have recently demonstrated this. What we have demonstrated is that by using and developing what we call ORS nano-IR, which is of resonance and short pulse and low power infrared nanospectroscopy, we could put our single protein between a gold tip and substrate nanogap. And by doing so, we could retrieve simultaneously morphological maps and IR absorption maps in the amide band 1 of single proteins, such as, for instance, thyroglobulin. Then what we could do, we could point our tip on single proteins and acquire fMIR spectra. And what we were interested in comparing this spectra with the ones obtained by FTIR on billions of proteins. And by doing a second derivative analysis of the spectra in the amide band 1, we could deconvolve the structural contribution and calculate the secondary structure of the protein. And we could demonstrate that we could calculate the secondary structure at the single molecule level with a similar accuracy as in bulk on billion on proteins. Then what we have been doing in this year, we have been using these technological advances to allow a series of scientific discoveries. And of course, we have been focusing on the problem of protein misfolding and aggregation, as well as liquid-liquid phase separation. And now this occurs in vitro, but also how it occurs in living organisms, and how these can be treated, for instance, by using small molecules to prevent the disaggregation. So I will show you in the last 5 minutes quickly some of the of our latest scientific discoveries in this field. And these, in this case, will indeed be focused on the problem of protein self-assembly. So what we know is that proteins, as I showed you before, can go from a dispersed phase to an oligomeric, protofibrillar, and fibrillar state. And this is what we called amyloids before. What we also know is that proteins can also undergo to another state, which is called liquid-liquid phase separation, where proteins form droplets that in the cells are used to compartmentalize biomolecules. What we wanted to do, we wanted to use fMIR and infrared nanospectroscopy to characterize this process in high detail. So now what we can do, if we acquire a morphology map of these aggregates, we can go on single aggregates one at a time. And by doing so, we can acquire IR spectra as well as the stiffness maps. And we could use that to see how during the aggregation of protein, there is this formation of cross beta sheet that is related to these amyloids, and it is related also to the disease. So we could use this approach to reconstruct the process of protein misfolding and aggregation at the single aggregate scale, at the nanoscale. Then we could do the same to study how these proteins can condensate into liquid-liquid phase separated droplets. So as I said to you before, normally these protein droplets are used in cells, for instance, to transport and compartmentalize material at their interior. However, if these proteins undergo a mutation, they can undergo a similar fibrillar aggregation. So this means that from a reversible droplet, we pass to some fibrillar-like irreversible aggregates, which are associated to the onset of amyotrophic lateral sclerosis. What we were interested in doing, we were interested in characterizing how the structure at the morphological and chemical level of these aggregates was changing in association to disease. And what we could demonstrate for the first time is that indeed, when there were disease-associated mutations in the proteins, we had a net increase of intermolecular antiparallel beta sheets, similarly as it occurs in Alzheimer's or Parkinson's disease. On the other hand, as I said to you before, this process of irreversible, very resistant protein aggregates can be exploited to produce functional materials. And for instance, in our recent studies, we just demonstrated that if we apply shear on these protein condensates, similarly as a spider will do in its groin to produce silk, we could go from a liquid-liquid phase separated state to a fibrillar one. And we were interested in characterizing how this process occurs at the molecular level. So what we did, we used nanopolarization infrared spectroscopy to study the anisotropy of these aggregates. And what we could understand is that going from the droplet to the fibrillar state, as expected, we had an increase of intermolecular beta sheet content. And in particular, what we could observe is that the fibrillar structure was dependent on the polarization of the IR light that we were using. And so this means that these fibers are highly organized materials. So we go from a dispersed phase to a droplet one, and then by applying shear, we can produce highly ordered fibers with beta sheet structure, which can be used as functional biomaterials. Finally, in the last couple of minutes, I will also show you how having this capability to characterize not only morphology, but also chemical properties at the nanoscale can be used to study molecular interactions. for instance, between these amyloid aggregates and a small molecule that can prevent Alzheimer's disease models in living organisms. So what we know is that normally protein aggregates, and this is indicated normally by a sigmoidal curve where there is a dye that is sensitive to the production of fibrils. And at the beginning, we have no fibrils, then the protein aggregates, and at the end, we reach a plateau phase where all the proteins are aggregated. In my previous lab in Cambridge, it was discovered that a molecule called bexarotene can delay this aggregation in vitro, but also in C. elegans models of Alzheimer's disease. So what this means that if we have control worms that naturally develop a model of Alzheimer's and they have protein aggregates in their muscles and brain, if they are treated with this small molecule, they do not develop aggregates, so they do not develop the phenotype of the disease. We were interested by using infrared nanospectroscopy in understanding how this small molecule bexarotene interacts with our aggregates. And to do so, of course, first we characterize the aggregates alone one at a time to obtain the infrared spectra, and then we did the same with the molecule. And we observed a striking difference in the spectra, which was indeed related to the presence of the molecule together with our aggregates. And as well as the characterization of secondary structure. Then I will not go too much in detail here, but please feel free to read the publication as well to make any question. We use artificial intelligence, so clustering and principal component analysis to understand how the molecule was interacting with our aggregates in comparison to the aggregates alone. And what we could conclude is that the molecule is interacting with our protein aggregates through single hydrogen bonding with the protein backbone. And so we could really study intermolecular interaction at the single bond level, at the nanoscale. And with this, I would like to thank you for your attention. And I hope I show you how, in order to study biomolecular processes which are very heterogeneous at the nanoscale, it's very useful to apply atomic force microscopy, but also combine methods to arrive at chemical properties. And I hope I show you how this can be useful to unravel the rules of life, understanding the concept of neurodegenerative disorders, and also maybe in the future to produce functional biomaterials. And with this, I'm done, and thank you very much for your attention.